Welcome to this tutorial introducing PHI Canto, the PHI-base Canto tool for author self-curation. My name is Elaine Cusick and I work at Rothamsted Research, where PHI-base, the Pathogen-Host Interactions Database, has been developed and is maintained. PHI-base contains manually created molecular and biological information on genes proven to affect the outcome of pathogen-host interactions. In this tutorial, I'm going to introduce you to PHI Canto and demonstrate the curation of an example publication using saved screenshots generated from the PHI Canto website. Canto is a community self-curation tool originally developed for the curation of literature documenting fish and yeast mutant phenotypes into PHI-base. You can see their website for further information. PHI Canto has been adapted from Canto for PHI-base to incorporate the multi-species data in PHI-base and enable authors to self-curate their data into PHI-base upon peer-reviewed publication. I'll run through the various data capture models for you. Canto captures data like this: gene ID details, gene annotation, such as a phenotype or a Go term, annotation extensions for additional details related to the annotation. In PHI-base, we are trying to capture the data like this: pathogen gene, host, pathogen-host interaction annotation. For PHI Canto, the tool has been configured to interpret the multi-species PHI-base data like this: pathogen gene annotation, annotation extensions specifying the host data. To get started with PHI Canto curation, you will need the PubMed ID of the publication to be curated and the UniProt IDs of the genes to be curated. I have selected this publication as an example. Fusarium graminearum gene deletion mutants, MAP1 and TRI5, reveal similarities and differences in the pathogenicity requirements to cause disease on Arabidopsis and wheat floral tissue. In this example, we will be curating data from one pathogen species, Fusarium graminearum, two pathogen genes, TRI5, encoding trichodiene synthase required for mycotoxin biosynthesis, and MAP1 encoding a MAP kinase, and two host species, Arabidopsis and wheat. We will generate 4 PHI phenotype annotations and 2 Go term annotations. I will also demonstrate the duplicate and edit functions to aid in speedy curation. Let's get started and see what PHI Canto looks like. Type in the PHI Canto web address or access it through the PHI-base website. So step 1 for curation is to type in the PubMed ID and click Find. Check the publication details displayed are for the expected paper and continue. For step 2, select Start Curating and enter the curator's name and email followed by Continue. Next is step 3, where we need to add the UniProt IDs for TRI5 and MAP1 genes to create a pathogen gene list. And continue. Confirm the correct pathogen genes have been pulled up from the UniProt IDs. More genes can be added if required or existing genes removed. Running through the contents for these headings from left to right, we have the entered UniProt ID under Systematic Identifier. Next to this we have the UniProt entry name ID, which PHI Canto uses as the gene name. Then we have synonyms, gene product information, and the pathogen organism in this example, Gibberellizia, is also known as Fusarium graminearum as described in the publication. Click to continue. We are brought to the PubMed ID summary page next, currently showing our entered pathogen genes on the left and the publication details on the right. It is possible to pause and reactivate curation when you see this button, or submit to curators once finished. For step 4, we shall select a pathogen gene to curate by clicking on the UniProt entry name, in this case for TRI5. This brings up the TRI5 gene page. So for step 5, we can choose a curation type for our first annotation. There are 8 different curation types available. 5 phenotype, effector gene, genetic interaction within a pathogen, physical interaction within a pathogen, Go molecular function, Go biological process, Go cellular component, and post-translational modification. Further details are available on our help pages. We are going to curate a 5 phenotype annotation. The Fusarium graminearum tri5 deletion mutant has been inoculated onto Arabidopsis floral tissue. We shall be annotating this pathogen-host interaction outcome using one of 9 upper-level 5 phenotypes. Firstly, we need to provide the allele details for the tri5 deletion mutant by clicking on single allele. You enter the UniProt entry name under allele name and select deletion from the drop-down menu for allele type. Note that PHI Canto now changes the allele name to include delta to indicate deletion. Select OK. A pathogen genotype has now been created for tri5 delta. To continue, click on Add a new 5 phenotype for this genotype link. And we move on to step 5C, adding an annotation term. In this case, to the curation type PHI phenotype. I am looking for unaffected pathogenicity in the search box, which is one of 9 upper-level 5 phenotype outcome terms used in the preliminary annotation. Subsequent additional 5 phenotype annotations can be made using lower-level terms such as unaltered spores and so on. Definitions for our 9 high-level phenotype outcomes are available in our publication here. Back to the annotation term. In some cases, it is possible to add child terms to your annotation term. In this case, there are none available. There is also an option to suggest new child terms if you wish by clicking on the link. Once ready to move on, click Proceed. For the next slide, we can choose an evidence code for our annotation term from the drop-down menu. In this example, the tri5 delta genotype resulted in unaffected pathogenicity as a 5 phenotype annotation term, and this was tested with a functional test in the host organism, Arabidopsis. Next, to add in additional details about the annotation using the available extension options. In this case, for tri5 delta functional test in host organism, with an annotated 5 phenotype of unaffected pathogenicity, we will add in information about the pathogen strain NCBI taxonomy ID, the pathogen strain name, the host tissue infected, the host NCBI taxonomy ID, and the host strain name. Some extensions are entered as free text and some from a drop-down menu. By clicking on the link for pathogen strain NCBI taxonomy ID, the details can be added to build up the annotation extensions. As the extensions are added, the information appears below. Next, adding in the Fusarium graminearum pathogen strain name, which is PH1, and the host tissue infected, which is the inflorescence, followed by the host NCBI taxonomy details, and host name and strain, which is Arabidopsis thaliana strain Landsberg erecta. Once we have entered all our annotation extensions, it looks like this with the blue arrow. These extensions can be edited by removing and re-entering as required. And proceed. There is an option to record any additional information that you feel may not have been captured in the annotation. And proceed. Here is a summary for the annotations made for the tri5 delta genotype. There is one annotation under 5 phenotype. A visual check can be made here and annotations can be edited, deleted or duplicated using the options on the right. At this point, the author may only be making one annotation and may wish to proceed to the final step 6 and submit to the curators by clicking back through the blue buttons to the PubMed ID summary page. However, we are going to make a new annotation using the speedy duplicate and edit options. Duplicate the annotation and select the edit option to create a new PHI phenotype annotation for the Tri5 delta genotype. By clicking on the Annotation Extension Edit button, we are going to change the experimental details to include a different host plant. Under Edit Extensions, we shall click the red cross on the experimental host ID and experimental host strain to delete the existing extensions. The new host NCBI taxonomy ID and host strain name can be added as before. In this case, we are changing from Arabidopsis to the wheat host Triticum F. Stephan strain Bob White. In addition, we can now enter another extension as Fusarium graminearum infection of wheat ears causes the disease ear blight. Next, we shall check the extensions added so far. These look OK, so we will click Continue. We need to enter a new PHI phenotype annotation term name. In this case, reduced virulence was seen when Tri5 delta was inoculated onto wheat ears. Here we can check through the edited annotation. In this case, the genotype stays the same. We have a new PHI phenotype annotation term, no change in the evidence code, and the annotation extensions have been amended to change the host details, and we have added in details on the disease caused. Select OK to continue. Now we see a summary of the two PHI phenotype annotations made for this genotype, Tri5 delta. The second annotation includes the new reduced virulence annotation term and host details. We click back to the genotypes. This shows that two annotations have now been made to the Tri5 delta pathogen genotype for this publication. Click back to the summary. Back at the PubMed ID summary page, we can select the Tri5 gene again to make a further annotation under a different curation type. We know from the publication that Tri5 encodes for a trichodiene synthase involved in mycotoxin biosynthesis. Under curation type, we shall select Go Biological Process to make a new annotation. Here we search for the correct annotation term, mycotoxin biosynthetic process. Select child terms if required, but not in this case. And then we proceed. Select an evidence code for this Go annotation. Here we have chosen inferred from a mutant phenotype and proceed. Add in the extensions, none in this case, or additional comments and proceed. Check the new Go annotation to the Tri5 gene looks correct. And then we go back to the PubMed ID summary page. There is another gene here from this publication to annotate. We do this by clicking on it. The steps can be followed through again. Without showing all the steps, I have created a MAP1 delta mutant single allele genotype and annotated two PHI phenotypes with evidence codes and relevant extensions. I have also added a Go molecular function annotation term with evidence code. These annotations can be seen here in the MAP1 gene summary page. Moving on to the final step 6 for the curation of this publication, all the annotations can be checked on the PubMed ID summary page. We have 2 genes, 4 PHI phenotype annotations, and 2 Go term annotations. This can be submitted to the curators now. Once the curation is finished, there is an option to add any further comments regarding the curation of the publication to the PHI Canto team. Finally, a summary appears acknowledging the curation of 2 genes for this publication. Post-submission or a pause in curation brings the author back to this page, where recent sessions are visible for quick access. It is important to note that PHI Canto can only have one assigned curator per PubMed ID to prevent duplication. However, each author can have several papers under curation at the same time. The author can find help by emailing the PHI Canto curation team or looking through the help documentation. Our vision is that a simple paper will take about 15 minutes to curate, and a more complex paper a bit longer, perhaps 30 to 45 minutes. Thank you for your interest in PHI Canto. I hope this has been a useful introduction for you. If you have any queries, please feel free to contact us by email at the below email address.